Hello, attendees who are joining us. Welcome. My name is Jacqueline Sarnice with Elevated STEM Teaching, here to do our first COVID-sciqueen investigation. I want to see which groups you represent. Are you a family? Are you an educator? Are you just an interested visitor? And that's okay if you are an interested visitor. I want everyone to be able to join us and participate in our sessions, whether you are someone who's interested in doing more of these investigations, or if you're trying to get your students or your children to do something at home with you right now. And I also want to know how old these children are that are with you, so I can make sure that I'm asking the appropriate questions. I'll tell you a little bit about me, and to start by saying that I'm an educator. I am not a technologist, but that's kind of what's happening to all of us now. We're trying to navigate how to remotely participate in work meetings or remotely participate in school. So we're all kind of learning to be technologists in an era where I guess it's probably pretty important that we all know these skills, but we all kind of got thrown into it without that much preparation. I'm a teacher by trade. I went to the University of Miami and I studied biology and secondary education. So I did my student teaching down in Miami. And then I moved to New Jersey, which is actually where I'm from, and I taught high school biology there. And I started a biomedical magnet program at that high school where I taught. So, and then I moved to California. And once I live in San Diego now. And I work with middle school science teachers. I work with a curriculum company, and I actually, as you see, I just started my own company right before all of COVID-19. So I'm trying to navigate what it's like in these uncertain times at the same time as you. But this has really familiarized myself with the next generation science standards. And the educators who are here with us should be pretty familiar with them. Those are the standards that teachers follow all throughout the country, in most of the states, just to make sure that we're all teaching the students the same amount of science content, and that we're making sure that all of our students are ready for this global economy. So I work mostly with middle and high school. I'm trying to learn kindergarten through 5th grade. This is a little bit new for me. So I'll give you access once this is over to our Google Drive folder, and it's going to have questions that are pertinent to students K through 12. The very first thing that you want to do, because I did not mention this in the directions, is if you did not take your strawberry out of the freezer, or if you are using frozen fruit, make sure you go ahead and take it out now. We don't want them to be frozen when we go ahead and mash them, so I want to give as much time as possible for it to thaw. You could probably even run it under the water in the sink if that's something that's accessible to you at that time. And I want to make sure that everyone has all of the materials that we're going to need. I went ahead and printed out my little directions. I am a little old school. So if you don't have a strawberry, you can use a banana, so I'm told. Maybe you have some bananas. I know that getting fresh produce is something that's difficult right now. So I thought maybe you'd be more likely to have some frozen berries in your freezer that you might want to work with. And if you have a fruit that's different than a strawberry or a banana or a kiwi, you go ahead and give that a try too. I'd love to hear how it works for you. You're going to need some dish soap, nothing fancy. This actually, I don't even really use this on my dishes anymore, but so I thought this would work great, and I tried it out and it does. Supposedly, you don't want that to be lemon flavored. I have to be honest, I don't know why, but maybe some of our high school students who are tuning in, maybe that's something you can look up afterwards, why lemon scented dish soap does not work. And also, you don't want it to be antimicrobial. If you have antimicrobial and that's all you have, give it a shot. We're going to go with salt. Any regular salt will work. You need a strainer of some sort. I've seen bigger versions of these really fine mesh strainers for pasta or something. That'll work fine too. It doesn't need to be this small. If you don't have a strainer and you have maybe cheesecloth, I've even seen people take t-shirts, an old piece of fabric that you're not using anymore, and you can cut it into little squares. We're going to use it to filter So anything that you can kind of get over the lip of a cup or a tube. You're going to need a test tube. So in my real science classroom, I would have plenty of test tubes for my students to use. I've been trying to think of things you could use at home, perhaps maybe a mason jar if you have one. You want to make sure that if you're going to have enough of a height. So if you are using a tube that's a bit wider than this, we'll probably just have to pour more material in it. And if you're working together as a family, perhaps we can put some, multiple of our strawberries together. You need some sort of sealable bag. If you have a Ziploc bag, that works fine. That's what I used to use in the classroom, especially if you have to get it to multiple people. I have a resealable, reusable sealable bag, just trying to be economically friendly in these times. Traditionally, you would use PPE, personal protective equipment, which might be something you keep hearing a lot in the news right now. Personal protective equipment, or PPE, those would be your gloves, perhaps you have your goggles, maybe even a lab apron. As I'm sure you know that I don't think you can hide from the news right now, but that our hospitals and our healthcare workers are in really, they're in need of gloves and they're in need of their PPE. So we're not going to be using them today. If you have any extra at home, perhaps you want to donate to a local hospital or a local healthcare center. We're not going to be working with any equipment that is going to really irritate you or anything today. You're going to need some sort of a long, skinny stick. When I did this the first time, I used a chopstick and it worked great. So if you have maybe leftover takeout chopsticks, maybe you're supporting your small businesses, you can go ahead and use that. I was trying to think last night because we did a deep clean of our kitchen this weekend and we accidentally threw away all of our chopsticks. But I found a thermometer that has a nice long skinny end. This will work great for us. If we were in the classroom, we'd be using a glass rod at this time. I don't have them at home, and I'm sure you probably don't either. You're gonna need a tablespoon and a teaspoon measuring spoon. If you don't have official ones, you could just use a small dining spoon as your teaspoon, or maybe a big one as your tablespoon. And I don't want to say measurement isn't important, because measurement is very important. But what we're doing today, it's not going to be super, super exact. So you can kind of guesstimate if you don't have that with you. You're going to need some water. If you have, I'm sure you don't have distilled water in your house, but if for some reason you do, awesome. I just took some Brita water just to make sure I wasn't getting it directly from the faucet, if that's an option. If you have some sort of distilled or filtered water that you can access, that would be super helpful. And did I forget anything? Oh, if you want a rubber band, if you are using a cheesecloth, or if you're using a piece of fabric and you don't want your guts to go everywhere, which might happen, you might want to use a rubber band because you can secure your filter on top of your tube. And if we have time, so we're going to be mostly extracting DNA from strawberries, but if we have time, I will do DNA extraction from our cheek cells. And that's what the Gatorade, I have it behind me. If you saw that in my post, I said optional Gatorade. If not, if you still want to participate, we can use salt. It's just a little grosser. So let's go ahead and get started. But before I start the actual experiment, I'm going to share my screen with you. So here's a question. These are for children and parents and educators, everyone who's involved in this. Can we think of some characteristics of living things? So what that means is, what does it mean to be alive? What must an organism have to be considered alive? Perhaps you can think back to class, just on a very basic level. Maybe you're thinking about plants and animals. Maybe you're a little bit at an advanced level, and we can think also about bacteria and other single-celled organisms. But what is it that makes them alive? Perhaps you thought about oxygen. Living things need oxygen to survive. Or perhaps you thought about food. They need some sort of nutrients in order to survive, whether that's actually eating or whether that's absorbing nutrients from the ground or from their environment. But there's one main particular characteristic of living things that I want you to focus on. and that is cells. Living things must be made of cells. And if you're a little one, maybe you haven't really talked about this before, but all of our bodies, every living thing is made of teeny, 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 tiny, microscopic cells that all work together. They have individual jobs, and then they can start working together as tissue, or maybe even once you get that further, tissue works together to be an organ. So something like your heart or your brain, all little cells all doing the same job. Now, what is the biggest topic that we're talking about right now? The coronavirus, right? That's why we're home. That's why we're learning how to do distance learning. What is a coronavirus? This is a much more complicated picture than you need to understand right now. I just want to get us doing something somewhat relevant. We keep talking about all of this in the news, but what does it really mean? And one of my very, very good friends, he is a very, very smart individual. We went to graduate school together at a prestigious university. And he told me that my COVID-sciqueen logo looked like a coronavirus cell. And that hurt me a little bit because coronaviruses do not have cells. In fact, viruses in general do not have cells. So at home, think to yourself, if a virus is not made of a cell, is it alive? Now, something that we need to really make sure that we're doing as educators and even as parents when we're asking questions, especially when it's through distance learning, is we want to enact wait time. And that's something that's actually really great that kind of happens automatically with distance learning, especially if you're not doing something live. But that's one of the main things that I've seen in classrooms when I get to go see my educators is that I see sometimes a lack of wait time. So sometimes we need to process a little bit. And if you've still been thinking about that question, the answer is no. Viruses are not alive. Now, if you see on the outside, there's all these little spikes. Those are proteins. Then it says lipid membrane, but a lipid is just a fancy word for fat. So basically it's a covering of fat with some spiky proteins. And in the middle, you have RNA, which might mean nothing to you. Maybe you've never heard of RNA before in your life, but maybe you've heard of DNA. Now, DNA is something in all of us. What have you heard of when you heard of DNA before? Maybe you heard about your genes. Maybe DNA makes you think of your parents because that's what your parents pass along to you. Your biological mother and your biological father pass on half of your DNA, so you get half from mom and half from dad. And not only do we have DNA, but all living things have DNA. But what I mostly want to get at is that viruses insert their DNA or their RNA into our cells to hijack it or to take it over. So if anything has DNA, then technically this virus can get inside and it can hijack your cells, and that is how you get the coronavirus. And you'll see in some of our questions that follow this session, I'm going to ask some of those questions. So how can we stop it? How can we stop the coronavirus from working? One of the main things I'm going to talk about today is the use of soap and how that's going to work in our experiment, and how using soap on your hands and your body parts can help stop the coronavirus itself. Depending on your grade level will be how much detail we're going to get into, but it will come full circle. So the whole idea we're talking about here is DNA, and we can expand on this in the next session, which is my hope. So I want to get ready to get started, and you're going to take your fruit. In a perfect world, everyone would have their own, but it's okay if that's not the case. Maybe you're all working together. I don't know what materials you have at home. I don't know how impatient some of the children that might be standing next to you are, but we're going to put our fruit in our bag. And if you're using something like a banana, perhaps you give every participant a little chunk. You want it to be big enough that you can kind of fit it in your hand. You don't want it to be too much bigger than that. If you're using a really, really huge bag, maybe you put them all together and everyone kind of works together. So we're putting our strawberry in our sealable bag. We're going to squish all the air out. Now, one thing I'm going to be doing, if some of you are still putting your strawberries in your bag at home, I have my pen and I printed my lab protocol, which if you are participating in this after the live stream, you'll have this available to you, which you can print if you'd like. And this is a really good practice I like to teach my students. Every time you do a step in the lab, you cross it out, and then you know what step you're on. Especially as an educator, if you're a science educator and all the students in a classroom are calling your name, for me, it'd be Miss Sarnees, Miss Sarnees, Miss Sarnees, I need help. Well, great, which step are you on? So if they cross them off, they know which step they're on, they know exactly what to ask for help. And I call it keep calm and follow the lab. It's a little outdated, but it was an original meme. Okay, so we have our strawberry, we get rid of the air. You can close it if you want. We're gonna open it again, but you're gonna squish it. Ooh, I don't think you can hear because I have my headphones in, but it's squishing really, really nice. You're gonna squish it in your hand, not too much yet. Now we're gonna add the extraction buffer. So there's a couple steps here. First, you're gonna need a tablespoon. This is the bigger one, your bigger spoon. You're gonna need a tablespoon of water and put it in your bag. So I have my water. This came from my Brita. So I got my tablespoon, I got my water. I'm going to go ahead and fill it up. Or you could always put this straight under the spigot. I'm gonna pour my water into my sealable bag with my strawberry. And now you need a few drops of dish soap. Let's see. Oop, that wasn't drops. That was a little bit of a spigot, but that's okay. This is not this is not that precise of an experiment. Some experiments, it really, really matters how precise you are. Most experiments, in fact, matter how precise you are. I picked this purposely because it's really hard to mess it up. Next, I'm going to add some salt. It says a pinch of salt. I do have a little tiny spoon that says pinch, but I'm just going to go like that. Maybe again. If you have meat tenderizer at home, especially if you're doing this without the live stream, you can add the meat tenderizer to this too. It supposedly helps break down the proteins that might get in here. So when I say DNA extraction, what I really mean is we're going to remove the DNA from everything else. So I got my strawberry in my bag. I added 3 things. I added water. I added a tablespoon of water. I added a few drops or a small spigot of dish soap. And I've added a pinch of salt. So we're going to seal it really good because now you're going to squish it for real. So we don't want to make a big mess in our kitchen. We're going to seal it and you're going to squish away. Now, my middle school and my high school students, you're going to have some questions about why we did certain things in the lab. So, strawberries. Are strawberries plants or animals? This is for our little kids too. Hopefully, you've said plants. Strawberries are a fruit. Fruits are a plant, part of a plant. And perhaps my middle school and high school students remember learning in class that plant cells have something different on the outside that animal cells don't have. What is that? What is the outermost covering of a plant cell? This is my wait time. Perhaps you remembered it was a cell wall. So when we're squishing, we are manually trying to break apart those cell walls. We want all those cells to separate as much as possible. You should be squishing for 1 minute. And hopefully my talking has been about 1 minute. If you have a few family members all doing this at the same time, maybe I'll give you a second to catch up. Now, while you're catching up, DNA inside all of our cells. So can you see a cell? This is for our little kids too. Can you see it, one single cell by itself? No, not most of them. There are some exceptions to this rule, but in most cases, no. You cannot see one individual strawberry cell or one individual human cell with your eyes. You need a microscope. So we call that microscopic. So if I'm going to be taking DNA out of these cells, how am I going to see it? DNA is 6 foot long. Oh, here are my periodic table pants that you can see. 6 foot long is how long our DNA is, but it's microscopic. It's so teeny, 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 tiny wide that you cannot see it by itself. So we're going to do something special to get all the DNA to clump together and get really, really, really stuck so that it gets wide enough that we can actually see it with our eyes. All right, so hopefully everyone has their DNA, not their DNA, their strawberries all squished inside the bag. I'm gonna mark off step 3 on my lab protocol and step 4. Okay, step 5 is telling me I need to strain this. So I wanna get the solids away from the liquids. My little kids, perhaps you remember that there are 3 types of matter. You have your solids, you have your liquids, and you have your gas. We want to get rid of the solids. I'm only interested in the liquids in this bag at this time. Okay, I'm going to be using my measuring cup because I don't have a funnel. If you have a funnel at home, or if you have something else with a pour spout, that would probably be the most helpful, because I want to get my strained contents into this teeny, teeny, tiny opening of a test tube. So I'm going to take my strainer and put it over my measuring cup. I'm going to take the contents of my bag. I'm going to try very hard to be very careful and not make a mess. And if you see, all the solids are staying in here in the strainer, and all my liquids are getting in the bottom of my cup, which is what I want. If you're using a cheesecloth, this would be a good time to put the cheesecloth on top with your rubber band, if that's possible, and you don't want to overflow. So I'm going to throw away my strawberries. And I want to get my liquid strawberry mixture into my mock test tube. If you have a funnel, this would be a great opportunity. Be really, really careful. I'm going to pour it in. And you want to make sure you're watching the side of the tube. You don't want to overflow it. We still have to add rubbing alcohol, which I apologize, I did not mention when I first went over the materials, because it needs to be ice, ice, ice cold. So if you, hopefully you saw the directions ahead of time and you have your ice cold rubbing alcohol waiting for you in the freezer. So I'm going to grab that when I'm ready. Just going to put a little bit more of my strawberry mixture into my tube because I want us to be able to see this really well. So if you're doing this with a few family members, maybe you need to get everyone else to catch up. I'm going to get my rubbing alcohol. I put the rubbing alcohol in a little mason jar because our rubbing alcohol bottle was pretty big, and we have a lot of things in our freezer right now because we don't want to run out of items amidst this quarantine, as I'm sure many of you are too. So I didn't want to take up too much room in my freezer. If you're ready to move on, you can. If not, I'll explain this again. I'm just gonna take a couple minutes, so I wanna make sure I get it ready. If you're ready with your rubbing alcohol, it has to be ice, 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 ice cold. And we're going to pour it in to our test tube at an angle. So perhaps a 45-degree angle. So I'm going to turn my tube on its side. And if you have a parent with you, if you're especially a younger child, perhaps you want a bit of help. Or maybe you can ask an older sibling. So if you have some of your high school students with you or the middle school students with you, they can help. And it wants to be about a tablespoon. We're guesstimating here. It's not that important. I'm just going to slowly pour it down the side. And I'm going to let this sit for about 3 minutes. I have my liquids in the bottom of my test tube. Remember, if you're using a larger container, perhaps you're using a mason jar. So you see how much wider a mason jar is than a test tube. This wouldn't be my ideal situation if you were using this, but you know, we gotta make do with what we have. If you are using a mason jar, you want to have a high enough height. So perhaps you can use more strawberries, or if some of you are working together, you can pour all of your mixtures, filter it all out, and then pour it into your mason jar. And the same would go with the alcohol. If you're using a mason jar, you probably want to go a little bit more than a tablespoon. So maybe a couple tablespoons here. Same idea though, holding the mason jar at a 45-degree angle and running the alcohol down the side. If you used something different, I'd love to hear about it, what you decided to use for your tall, skinny container. So for your test tube, and I'd love to hear what you're going to use for your glass rod. I have my thermometer, a chopstick, works great. That's what I used before. A popsicle stick, though, again, you might find a problem. Your popsicle stick might not fit in here. I've thought about trying the side of a fork, so using one prong of a fork if you can get it in there. Love to hear what you're using. And this is a great way to use your engineering brain. Bamboo skewer, someone commented, great idea. So maybe you skewer at home, or maybe you have metal skewers that you put on your grill. Those would be great options here too. You just want something skinny enough that's gonna fit in here, and we're gonna pick up the DNA. If you recall, I mentioned that you have this lipid membrane or lipid covering, which just basically means fat. Now, you may not know, but soap is also made of a lipid. So like attracts like. And also like dissolves like. So that's why if you put salt in water, water will accept salt. They are both similar types of molecules. So dish soap, a lipid, or any type of soap, will be able to dissolve the membrane of a coronavirus. Similarly, in strawberry cells or human cells, our cell membrane is also made of lipids. So soap will break down cell membranes and this particular virus membrane. Not all viruses have that lipid membrane. So it's been about 3 minutes. And if you look here, you should start to see, oh, it's a little foggy because it was so cold. You should start to see some things forming. So this was all liquid. My little kids, my K through 5 students, my solid liquid gas, these were all liquids, but maybe you see some solids starting to form. And this is happening right where my strawberry mixture meets my alcohol mixture. You should see it right around here. Maybe you see some bubbling. Maybe you see, ooh, here's a really good piece. That is DNA. So we can actually go ahead and pick that up. So if you have your skewer, your glass rod, your chopstick, I'm gonna stick it inside here, right around where my strawberry mixture meets my alcohol. I'm gonna swirl it around a little bit and I'm gonna take it out. Oh, that's DNA. What does it look like to you? When I was teaching in the classroom, my students used to say this looked like snot or boogers. It's not though, it's DNA. So this is a lot, right? It's microscopic. Normally it's so thin you cannot see it with your normal eye, but we got so many little pieces clumped together that we're able to see it. My middle school and high school students, we as humans are called diploid, meaning we have 2 copies of all of our DNA, all of our chromosomes. We get one from our mother, our biological mother, and we get our second copy from our father, our biological fathers. Strawberries are great for DNA extractions because they're octoploid. So initially I said diploid, meaning 2, di meaning 2 copies. This is octoploid. Think about an octagon. What does oct mean? 8. 8 should mean, so this is 8 copies. So that's why DNA is such a good, strawberries are such a good example for this experiment because they have so much DNA inside their cells. And you can keep going ahead and picking up more. Don't eat this though, or drink this. I hate that that has to be something that is said. My students in my classroom in high school used to ask me every single experiment, can I eat this? Can I drink this? And I love them to death. I'm still in touch with many of them. A lot of them pursuing STEM careers in college right now. I have students at Stanford, I have students at Villanova, Princeton, all of these very intelligent humans, but all ask me if they can eat and or drink the lab materials on a regular basis. So please don't. That's one thing with doing science experiments at home is we don't have that sort of mentorship, but hopefully that all of these materials are safe enough. If we have a microscope, I have a question. It says, if we have a microscope, we'll be able to see the cell. Wow, that's a great question. And yes, but here's what you'll want to do. These are very freezer burnt. This is why it's a great option to use these in your freezer. Perhaps you have a fresh one, or you want to wait until it's not freezer burnt anymore, but you want to slice it. So an adult or a high school student with decent knife experience would want to take this on, but you can go ahead and slice it really, really, really thin. You want to try to get the strawberry slice as thin as possible because it is going to be red. And if it's a light microscope, you want the light to pass through the tissue. So getting it really, really, really thin. I'm assuming if you have a microscope that you have microscope slides. I'll draw you a little picture. They're probably about yay big. And you could put your strawberry slice in the center of your microscope slide. If you have cover slips, that would be ideal, but if you don't have cover slips, that's probably fine, but you can't get really, really close. You don't want the microscope to touch your sample. So you wanna make sure they don't touch. If you do want it to touch, you would use something called a cover slip. And that's gonna be a small, thin square, also clear. So the slide will be clear and the cover slip will be clear. But you'll put a drop of water. So it's called making a wet mount. You'd put your little drop of water on your slide, and then you would put your cover slip on top. So you're making a nice tight seal, slide, strawberry sample, cover slip with some water in between. And then you could go ahead and look at your, look at the cells on the microscope slide. That's a great question. They can also see cells in the thin skin from an onion slice. Actually, yes, that's what I used to use. Onion slices are great. I haven't done garlic before, but I've heard garlic works really great too. I've even done potato. Potato, you need to be really careful, making sure you get those real thin slices. All of those things, the garlic, the onions, and the potato, they're all root vegetables. So that's something you might want to compare to a strawberry, which is going to be a fruit. It's a different part of the plant. Root vegetables grow under the ground, so they are not going to have light. And for my middle school, my high school students, there's one part of a plant cell that requires light, and it uses the light in order to feed the plants. So you will not find them. It's going to be green. You will not find them in garlic, onions, or potatoes. Excellent. In this particular example, I used the idea of the DNA extraction because I knew that that was easy. I knew that I could just go ahead and use materials that you probably had at home. As a teacher, I used to find myself making 5 a.m. Walmart runs before every lab, I feel like. If you teach other subjects, perhaps you don't realize this with science teachers, that they probably have to go out and buy their materials a lot in order to do these experiments. And oftentimes that will be at, you know, a Ralph's or a Vaughn's or a Walmart. It's not going to always be science grade materials that you're purchasing. So I knew that this was something easy that you had the materials for, and then I worked backwards. So I thought about how can I make this appropriate to the kindergarten standards, or how can I make this appropriate to the 3rd through 5th grade standards, and I kind of tried to mold it that way. With the little kids, the kindergartners and the 3rd through 5th grades, the standards are going to be on the physical science side, so that's why I was talking about it's microscopic, that the DNA and the cells are so microscopic, but if you put them together, you're able to see it. So that's how I'm making it appropriate to those standards. So they don't really get into DNA necessarily in K through 5. You don't really get introduced to that until 6th grade. So I had to think about other things that I could use to engage those younger students. It's 9:45. Thank you so much for attending. I want to do these at least twice a week. So I'm hoping to have a second investigation on Wednesday or Thursday. You will see them on all my socials. So you follow me on or like my page on Facebook, covid-sciqueen. On Twitter, you can search covid-sciqueen, but the actual @ is @elevatedstem. And on Twitter, you can look up on Instagram, you can look on covid-sciqueen as well. And I'm going to give you access to this Google Doc folder. So this is going to be the Google Drive folder that you will have access to. You won't have these other two things. Sorry. Investigation number 1, DNA extraction. And you will find the K through 2. So these are kindergarten through 2nd grade. These will be your 3rd grade through 5th grade. This is your middle school and high school. I did combine them because the standards overlap quite a bit. You'll see at the top that I listed the standards. If your family is from home, that's not going to be that important to you. If you're a teacher, perhaps that's a bit important to you, but at least it's just giving you some justification that I didn't make these questions out of nowhere. They are aligned to a national movement. So there's some questions. These are going to be for middle school and high school. But once you get down to the bottom, there's going to be a question that this is really meant for the high school audience. It's addressing part of a standard that's not really addressed in middle school. If you're a middle school student and you want to give it a try, I invite you to do so. And most of them will have an extension activity. It's probably a bit higher than the grade level or what's expected of them, but it might be relating it to something relevant in society today and maybe just making it a bit more exciting at home after you're finished the actual lab. Remember that I'm creating these questions and this content for a broad audience. So this might not be specific to your students, especially if you're an educator, or your children if you're a parent, but feel free to make those adjustments as needed. This is going to be a view-only access to this folder. So meaning if you are going to make adjustments, you'll need to make a copy of it. And then once you make a copy, you can make any change that you want. So here is the link. And I just added it into the chat. So that's going to be the access to the Google Drive folder. And you should also get access to a post survey. I'm not sure if it'll come to you right at the conclusion of this webinar or if it'll come to you in your email, but I would really appreciate if you took the time to fill it out and tell me what you thought. If you have any suggestions for moving forward, or you have any comments on how I can make this better, I would love to hear from you. Thanks again for participating. I hope to see you all again. Stay safe, stay healthy. and let's try to keep this educational. You know, we don't need to get our watching TV all day, every day. I get into those drives too. So let's keep investigating science. Have a good one, everyone.